Garhima, you have 15 minutes.
Good afternoon, everyone. Am I audible? Yeah. So, I'm a PhD scholar at THSTI under the supervision of Samrat Chatterjee. So, I really thank the organizers of the symposium for giving me this wonderful opportunity for presenting my work here. So, the title of my talk today is Development of Metabolic Model for Drug Repurposing, a Novel Approach for Precision Therapeutics. So before going deep into this study, I would like to give a brief background of what are genome scale metabolic models. So genome scale metabolic models are basically mathematically structured metabolic reactions and metabolites and genes. They are curated together in a mathematical format. So they are like valuable in silico tools which are basically used to study the metabolism of different organisms and under different disease conditions. So this slide basically sums up the different steps which are generally utilized during genome scale metabolic modeling. So first the omics data that we get, So the omics data that we get here is integrated into the model with the help of different model extraction methods, and then the flux evaluation is done. And after flux evaluation, we have biological utilities such as we can really find out the phenotypic differences between the disease conditions. We can look for the drug targets, we can look for the biomarkers, and we can also perform drug repurposing, and we can work with the personalized medicines. So now, since genome scale metabolic models are used in variety of diseases, so I'll be focusing here for cancer. So cancer is generally seen as a disease of uncontrolled proliferation. However, dysregulated metabolism or metabolic reprogramming is one of the major hallmark of cancers. So because cancer cells, they try to get more energy for their proliferation, so they try to perturb the metabolism of a normal cell from the normal metabolism towards the dysregulated one. That is the Warburg effect, which is generally seen in cancer cells. So in this study, I will mainly focus on the ovarian cancer. So in ovarian cancer, what happens is that the cancer cells from the primary tumor, they start losing the cell-to-cell addition, and they float into the peritoneal cavity where they start forming the spheroids. And these spheroids then further cluster together to make bigger spheroids, and then they start moving towards the secondary tumor, secondary sites where they form the secondary tumors. So now I would like to mention that this study is in collaboration with Dr. Ramre Bhatt and his team from ISC Bangalore. So they made a very fantastic observation where they have used OVCAD3 cell line, and they have shown that how ovarian cancer cell lines transit from a single layer, and from there, when they are cultured for 24 hours, they make the spheroids. And these spheroids are further, when cultured, they make bigger spheroids with a lumen-like structure. That means they have a central cavity. So the idea of this paper was to investigate what all the metabolic perturbations these cancer cells are going through, so that why they are transitioning from single cells to moduloids and to blastoids. So this was the aim of this paper. So we got this data from them, the proteomics data, and from here we filtered out the metabolic data, and using this metabolic data, we integrated this metabolic data onto the human genome scale metabolic model, which is Recon 3D we have used here. So this is a generic metabolic model for humans. It has mathematical curation of all the metabolic reactions which take place in a cell, and after overlaying it, we reuse different metabolic extraction methods like IMET, INET, and FASTCO. These methods are generally model extraction methods. We do not use objective function. Now, what does an objective function mean here? So basically, these genome scale metabolic models, they are in mathematical setting, they have infinite solutions. So we cannot really have a unique solution to this. So therefore, we need an objective function to get a solution to it. However, we have used these metabolic extraction methods. They do not really need any objective function prior for making a genome scale metabolic model, context specific metabolic model. However, they use threshold values to actually extract the subset of a metabolic model from the genome, the huge like generic metabolic model. So for each, if I go back in this, we had like 3 transitions. So we made metabolic model for all the 3 morphological stages, and using different thresholds and different methods, we had 18 context-specific models. Now these context-specific models were further evaluated on the basis of metabolic functionality score, like how much they are connected and how much they are metabolic functional. So we observed that IMET was one of the metabolic extraction method which showed the highest metabolic functionality score among all the morphological stages, and also we have used 2 different thresholds. It was the metabolic functionality score was robust for both the scores. We also did PC analysis to ensure whether the flux values which we got from this IMET models, are they clustering together or not. So we observed that these flux values from these 2 models were actually clustering together better. Now these models were further taken into flux evaluation. As I've already mentioned that these models have infinite solutions, so we really need something to work with. So for that, in that case, we did the flux sampling, and from there we got a solution space, and we found the optimal flux solution, the solution which is showing the maximum correlation with the input data that we have used while overlaying towards the genome scale metabolic model. So that flux value was taken as an optimal flux solution. Now for each morphological stage we had the flux profiles. Now we really wanted to see how these reactions are changing throughout the transitions. So for that we found the perturbed reactions. That means the reactions which are perturbed, either upregulated or downregulated when they are transitioning from monolayer to moraloid or moraloid to blastoid. So we observed that there were around 675 reactions which were perturbed in both the transitions. Now these 675 reactions, they are of interest because the genes which are involved in these reactions are basically involved in the perturbation. So using the genes which are involved in the 675 reactions, we performed the in silico gene knockout. So now these genes can be essential for normal cell growth. So first we filtered out the genes which were essential for the normal cell growth. For that we did a normal model, ovarian model construction, and we obtained the samples from GTX database. We again overlaid this data over the Recon 3D, and we made 166 normal model. And then we started knocking out in silico one by one in these models, and we saw that which all genes are basically reducing the growth rate. So we removed those genes, and we worked with those genes which were not really decreasing the growth rate of normal cells. So this slide basically shows the hits we got while doing in silico gene knockout. Now how we performed in silico gene knockout, suppose if there is a gene and it is catalyzing few reactions, so the bounds of those reactions were set to 0, and then the flux profile were evaluated. Now, so we wanted to see if the gene knockout is actually transitioning the, suppose a flux, I mean like for transition 1 we have monolayer and moraloid. So in moraloid we will put flux values, sorry, the bounds to 0, and then we again find the flux values, and if the correlation of that gene knockout model is higher with the initial stage model, which is here is monolayer, then we say that that gene was actually capable of reverting back the transition. So these were the hits that we got for transition 1 and transition 2. Even though if you look here, the correlation was not really very high, but the initial correlation between the flux profile was around 0.02 and 0.03. So comparing to it, and we have also searched earlier literatures, they show that this correlation was was enough to say that the transition was reverting back. Now, since we have seen the targets, now we really wanted to see what are the drugs that are available in market to repurpose it. So for this we did drug repurposing, and we exhausted drug bank database. From there we filtered out drugs which were basically targeting the genes which were reverting back the transition, and from there we got the drugs which were showing reversal, and for each transition 1 and transition 2. And there were few common single targeted drugs, And oseltamivir was one of the drug which was targeting NU1, which is a neuraminidase inhibitor. And this oseltamivir is generally used for influenza A and B virus. And we observed that this oseltamivir was majorly involved in many perturbed pathways, and it was catalyzing many reactions. So we chose this oseltamivir for our further in vitro validation. So we did in vitro validation using oseltamivir. Here what we did was that the spheroids, the control spheroids, so what we did was that control were cultured and they made spheroids, while when the control were treated with 250 micromolar oseltamivir, there was disintegration of spheroids. This was similar case was observed for the transition 2 as well. And we also looked whether this disintegration is a result of cytotoxic effect of oseltamivir or not. So we observed that in both the transition, even though disintegration was happening, but there was no cell death observed in both the transition. We further looked whether is there any effect of oseltamivir on non-cancerous cell viability. For that we took the untransformed mesothelial cell line MET5A, and we observed that there was no cell death. It was a little bit of cell death was there, but that was not very significant. So from here we concluded that this oseltamivir is a potential combination, can be given in a potential combination therapy for chemotherapy. And we also checked this oseltamivir in other 2 ovarian cancer cell lines, SKOV3 and COV362, and the similar results we got. So basically in this study, we have introduced a novel computational framework which utilized metabolic modeling approaches to identify mechanistic association between the metabolism and the metastatic morphological transitions in ovarian cancer. The effectiveness of oseltamivir, which is a NU1 inhibitor, in inhibiting spheroid formation in 3 ovarian cancer cell lines without inducing cytotoxicity in untransformed mesothelial cells. And this integrated approach can be used as a potential computational modeling in guiding drug repurposing efforts towards more effective therapeutic interventions. This paper has been published recently in iScience. So the future direction for this study is that since we have done in vitro study, we would like to take this towards in vivo experiments, and till now we have seen the metabolic changes in ovarian cancer cells. We would like to see what all metabolic changes are there in the untransformed fallopian cells and their transformed counterparts. And since we have done the experimental validation of a single drug, so there were other hits that we found, so the experimental validation of those drugs that we are planning to do. So lastly, I would like to acknowledge my supervisor, Dr. Samrat Chatterjee, the co-authors of the study, Dr. Ramre Bhatt and his team, my lab members, and THSTI for funding. Thank you.
Thank you. So we will take up questions.
Hi, thank you so much for the very interesting talk. I really enjoyed the genome scale metabolic model approach that you've taken. I just wanted a bit more information on the metabolic functionality scores that you had mentioned. Could you just shed a bit of light?
So these are like 56 metabolites which are essential for cancer cell growth.
Okay.
So we have considered those reactions. So what we did was we added a sync reaction every time for each metabolite, and then we again did FB analysis, and if the flux value of that sync reaction was higher than some threshold, then we considered that that particular metabolite was forming. So we said, okay, that is metabolic functional in this particular.
Thank
you. Thank you for that nice talk. I think you can answer, since I also work in metabolic modeling. So I want to know, like, can this IMAT algorithm you said, can it integrate all multi-omics data, like metabolomics, transcriptomics, and like fluxomics data?
So I've never worked with metabolomics data in genome scale. I think transcriptomics and proteomics we can really put with IMAT.
Okay, and can you use it in Cobra Pi, the IMAT algorithm? Can you use it in the Python-based Cobra interface?
So this study was done like entirely on MATLAB. I'm not very familiar with Python.
Okay. Thank you.
Hi. have we benchmarked our drugs that, for example, we have taken Alstromavir. Have we benchmarked with any known drugs and its work? Like right now we only have this results of one drug, right? And you have also said that we have screened drug bank, entire drug bank. Like what were the parameters that you found the key we are going to choose these drugs for validation only?
So if suppose I'm seeing the effect of a drug in my model, so the target of that drug should be present in the model. Otherwise, how can I see the effect of drug? So the perturbed reactions, the genes which were catalyzing those perturbed reactions, so we looked for the drugs which were targeting those enzymes basically. So this was the method we used for filtering out the drugs from drug bank.
But like, have we compared it with anything before?
Anything as in?
Like the known drugs for those targets, have you compared it?
So anyone has a drug, oseltamivir, yeah.
Have we compared it with the previously known drugs or
So
inhibitors?
this is a FDA approved drug only, oseltamivir for anyone.
Okay.
So, Garhima, I just was asking you one question, maybe I have missed it out. So, you said that you did some knockout, in silico knockout. So, how did you do that?
Suppose if there is a gene and it is catalyzing few reactions in the model. So I'll just fix the bounds of those reactions as 0. That means there is no flux going through those reactions. And then again that flux profile was evaluated. So to see if the flux profiles are correlating better with the initial stage flux profiles or not.
Okay. Another thing is, did you check it on multiple cancer cell lines? I mean, ovarian cancer cell lines.
Yes. So one was OVCAR3, the other two is SKOV3 and OV360.
So, OVCAR3 was able to culture properly. I mean, there are certain issues with these. I mean, SCOV3 is okay, but there are certain issues with OVCAR3 because, it takes time to grow and all these things are there.
So I may not be a good person to answer
Okay,
this.
I got it.
Since it is in collaboration. So yeah.
Okay. Okay, so let's thank the speaker then. So we have come to the end of this session. I would like to hand over the microphone to the organizers.